Yes, sir. So you are joining, sir? Okay, sir. Okay. Krishna, did you take one page and pen? आपने मेरा पेज ले लिया. Yes, Dr. Swati, we are just waiting for our HOD to join and we can start. Yeah, he's here.
Okay, no problem.
Yes, good evening, sir. Professor Ramakrishna?
Yes, good afternoon, everyone.
Yes, sir, we can start, sir.
Yes, sure.
Yeah. So, a very good evening to all my fellow colleagues, the students and the science aspirants, and warm good morning to Dr. Swati Dadhich. With this, we have to admit in between, so just sorry for this break. We are here for the first talk of the series, The Possibilities for a Science Enthusiast. And we have with us Dr. Swati Dadhich. Before she starts with her, talk, the first talk of the series on being a biology researcher to a medical communicator and writer, I would request our HOD, Professor Ramakrishna Huzurika, to say a few words about the background and the theme of the whole series per se. Please, sir.
So good evening, everyone, and good morning in the US. Today we have a talk by Dr. Swati Dadhich. So I was looking at her CV. So as she did PhD from JNU and then postdoc for a few years. So this is the path that most of us have taken after our PhD. But then she diverged from there. After that postdoc, then she started doing this medical writing and communication, working in different places. So this will be an interesting and a different talk for most of us who are biologists here. So especially for students, to see that there are other career options other than doing postdoc or becoming a teacher or working in a company. So I know a few people who have taken this medical, not only medical writing, but they have become working in patent offices or working in publishing houses, which are something alternative kind of options available for us. So it will be interesting for us to know that we don't have to stick only to one thing, but wherever your interest takes. I think that is the key. What you love to do, you should do it. Okay. Don't have to think about what people will think, other people, what they will think about you. If you are a professor, you may be getting this much respect. In other, you may not be getting. So all that those are all old days, I think. These recent days, everyone encourages, even parents, if you see, they encourage their children to do what they like to do. Rather than, if you see 30, 40 years back, parents used to tell us, okay, you become a doctor or engineer, whatever. Even if you don't like, we have to go that way. Okay. So things have changed. So this will be a really enlightening kind of talk for us, for all of us, including the students especially. So please listen carefully, and then as you complete your MSc, by that time you should at least know what you want to do and pursue it single-handedly. Or you can also change, as Dr. Swati changed. Okay, you went through here, and then what is... But important thing is first figure out what different options available. I think I will stop here and hand over to Dr. Manju.
Thank you very much, sir. I would just like to convey the message to all the masters and the PhD students out there as audience that the entire theme of this series revolves around talking of building up your future plans based on your basic training in masters and PhD. So that basic training as a biologist becomes the base of what possibly many more things you can do in the scientific domain, right? So whatever we will be talking in this series, basically it will be representing the extension of your basic training as a biologist. So that is very crucial, and the whole purpose of putting this series together is to sort of motivate and make you full of enthusiasm regarding whatever training you are getting at masters or PhD. So that would basically make bigger things out later, out of your life, out of your career, right? So with this, I would give a brief introduction about our first speaker, Dr. Swati Dadhich Mandal. So she's a scientist by training, and she's a medical communicator and writer by her passion. As she moved ahead, even though she has been doing great in her biology research, and I know her as a great manager, since the kind of work she is involved right now involves multiple activities, scanning the clinical trials, the agreements, supporting the investigators at National Cancer Institute in the capacity of senior regulatory medical writer, and her area of expertise in terms of the scientific domain comprises immuno-oncology, neuroscience, and infectious diseases. And you can see she did a technology transfer in her tenure also. She sort of spent her time into at National Institute of Neurological Disorders and Stroke, NIH. She did a postdoc in cancer biology at National Institute of Dental and Craniofacial Research at NIH. And there are many masters and PhD students as audience, she did her master's from Devi Ahilya Indore, and pursued her PhD from JNU Life Sciences, and also had a adjunct faculty position for a year or two. Apart from that, I would like to add that she is very much involved into different STEM programs that basically involves the scientific technology, the engineering, and the mathematical components, so as to to be put them together as a cohesive learning paradigm in a very interdisciplinary approach, which is the need of the hour, right? So with this little introduction, and definitely there are a lot of laurels to her in terms of the kind of publications she has in her scientific research, and many conferences like Gordon Research and all, we all know. So she has acclaimed the credibility of her work as a biologist, as well as the further extension of herself as a biologist in more areas we will be discussing more. Yeah, Swati. So Dr. Ravindesh, you can maybe get out of the presentation mode, and Dr. Swati, you please come up with your presentation.
Thank you so much, first of all, Dr. Ramakrishna and Dr. Manju, to give me an opportunity to share my journey with everyone, and especially the students who are very dear to my heart. I feel like I was there 10, 15 years back, and whatever advice I can share with them would be valuable, at least from my perspective. So let me go ahead and start sharing my screen.
Yeah.
Do you see it now?
Yes, Dr. Swati, it's there.
Okay, great.
Yeah.
So, hello everyone again, and I think it's 5 p.m. evening in Saturday, so you are spending time here listening me. That is very that itself shows that you are quite motivated about your career. So this is NIH, as you can see in the picture, and I have spent my last 10 years here. This is the National Institutes of Health. 27 different institutes are here in this campus at Bethesda, Maryland, USA. And I have worked as a senior medical writer past 3 years at National Cancer Institute, which is the biggest institute here. I just wanted to put the disclaimer because, the views and opinions expressed in this PowerPoint slides are myself, and they doesn't attribute to any government agency, its director, officers, employees, volunteers, members, chapters, councils, or affiliates, or any organization with which I am employed or affiliated. This presentation also does not serve as guidance of any agency of US government, nor does it represent the NIH and FDA's current thinking on this topic. This is just my personal opinions. So one more thing, I would encourage people to chime in when they have any questions. When something is not clear, please feel free to have questions ready. Even if we are taking them in the end of the talk, please get them ready for me. So just for giving you an outline of my talk, I will briefly go through my academic background, and I think that's important. Dr. Manju has really, given a very good background, but I think from a student perspective, I would like to give you where I come from and how did I make this journey so far. Then I would go with some highlights of my PhD and postdoctoral scientific work with you. I will share then the skills and capabilities to excel in medical communications, how you can become a medical communicator, writer, if you want to, and what are the resources which you can still start right now, where you are. You can keep building those skills. Writing skills are important for every profession. Doesn't matter whether you are a scientist, whether you are a writer, or like at any profession, writing and communication, verbal and written communication, both are equally important. So let me start with my academic background. I come from a very humble background, and my family is from Jaipur, Rajasthan, not far from you, Haryana. So I did my bachelor's in microbiology, industrial microbiology, as a specialization from Rajasthan University. There is a Maharani's College, a very famous college in Jaipur, and I did industrial microbiology as a specialization. And the reason why I'm putting it is because if there is an opportunity to take something which is like bench to bedside or kind of an industrial implication, it is important to have that kind of area or subject, because then you get an insight how the things we do in the lab can be implemented further. For example, when I took industrial microbiology, I learned how to do fermentation, how those fermentation products we use in our daily life, such as dairy products, beer production, etc. We also got an opportunity to visit those plants in like Ahmedabad, in Gujarat, different places, Amul plants, and that was kind of very inspiring to me. That what we do is not going, is really implemented in our daily lives. So that's where my bachelor's was. The master's was in life sciences, Devi Ahilya University, Indore. And This was through a common entrance exam JNU has for different universities, and I opted and got an opportunity to be here. The very good part of this program was that the teachers who came from different, outside, like abroad, after finishing their postdoc, were teaching us. So they were teaching us like a molecular biology skills, as you guys are fortunate about it. In your department, people coming from, after finishing their postdocs, they are coming here, they are teaching you all these cool technologies. And those are very important as a master students to, even though if you have an like kind of a scientific lab experiments, to master those techniques, even if it's like a digestion experiment or running a gel or developing a blot, it's very important to get those things done very good. And I did, and I was like kind of, I'm putting it just for the students' perspective, not like bragging myself, but I did secure 5 years funding for my PhD through in my master's itself. So there is, there is a NET exam. And that is a very important exam people do not only for teaching, but also to secure funding for PhD. I didn't want it to be dependent on my parents for money, for my PhD. So I said, well, I would first let get that clear. And that gave me a confidence and kind of boost to apply for the PhD program. Now here it is. If you want to have PhD, and many of you must be thinking about it, whether we should really do PhD or master's is enough. If you ask me, or if you ask anybody who has done PhD, I would say go for it if you really want to excel in your career. Education is not just important to build the skills, but to build your personality as well. And PhD gives you both advantages. It teaches you more than just get the research done. It teaches you accountability. It teaches you credibility. It teaches you tenacity. You never give up in PhD. You keep on working on your research project, and you keep on working what you really want to, get out of it. So there are sometimes adverse circumstances, but you go above and beyond to achieve your goals and objectives. So I am in favor of PhD by all means. One should do PhD, and PhD is passion. I'd like people say, okay, PhD is like a philosophy, like doctor in philosophy. I define it differently. For me, P stands for your passion, H stands for your hunger, and D stands for your drive. And that makes your PhD done in 5 years or 6 years. And when if personally somebody asks me, PhD gives you an added advantage in any alternative career as well. In US currently, 90% of the PhDs are doing something else, not academia, but they have added advantage in their career if they have a PhD. So I did PhD from Jawaharlal Nehru University, as Dr. Manju said, and I did in research infectious diseases, leishmaniasis, did publish well, had a lot of conference presentations, and that's a very important thing as well, to brush up your verbal and verbal communication skills. So when you get an opportunity to present your work, don't shy. Present it, make an oral poster presentation. And I did get opportunity to visit US at that time, 2 times actually, one in Gordon Research Conference and one in ASTMH, American Society of Tropical Medicine and Hygiene. And I was an oral presenter, and it was nerve-wracking for me as a PhD, like a very early student, to present before like great scientists of US, 250 in the hall. But, it just gives you the confidence boost later on in your career. When I submitted my PhD, I got a 6 months kind of break, right? Because when you submit the PhD, it goes to the 2 reviewers who review your PhD thesis. And at that time, I thought, well, what I can do, like other than finishing my papers in the lab, I joined Gargi College, Delhi University as an adjunct faculty. And if I would not have joined the college, I would not have known that how passionate I am for teaching and mentoring students. Because that, even though it was a short span, I guess 8 or 9 months, it just was very enriching experience for me to teach students, to mentor them, like what they want to be in their career, to scientifically demonstrate them how, the molecular biology techniques work. So I would say that was a very good time spent. Then I joined in February 2011. I came to this country, United States, and joined NIH. So as I said, NIH has 27 different institutes, right? And how we remember those, just because they all are by different body part names. For example, heart, there is a Heart and Lung Institute. Eye, there is an Eye Institute. I did my postdoc from Dental and Craniofacial Institute. And it doesn't matter what institute it is, but most of them does the cutting edge research in neuroscience, in cancer biology, in infectious disease. And then I did in cancer biology my postdoctoral research. So I made a pivot here. I did a PhD in infectious diseases, but then I wanted to grow beyond that. So I took the cancer biology for my research and postdoc. Did publish well. Again, I would say I would emphasize on, the publications are not like, you want to just publish whatever you get, but there has to be a thinking process, what projects you want to have, whether they have an could be, they can have an implication in the further research, or, it just motivates you to do your best each day. Like I remember working in the lab all the time, finishing my paper, and I had a very good PNAS paper within 2 years of the postdoc. So that was kind of again, I would say a confidence booster that I can do well as a scientist. So then what happened, and I will go further in my slides about that, I pivot again to become a senior, started with a regulatory medical writer at NCI, and then recently promoted to senior medical writer. So I would go with that later, but I just wanted to give you a little brief about this thing, and wanted to tell you that from a girl whose family has not been outside Jaipur, not yet, actually made her path from Jaipur to Indore to Delhi to US. So if I can do it, you can do it as well. So here it comes, the PhD just student, right? So I as a PhD student joined the lab of Professor Rentala Madhubala and in JNU. So in what was my kind of PhD objective? My objective was to identify the potential biomarkers for antimony drug resistance and development of novel compounds for the treatment of visceral leishmaniasis. I can imagine you have Dr. Manju as a faculty, so you guys are very familiar with leishmaniasis. So I'm not going to have much detail about this disease, but just a brief glimpse that I did work on a lot of clinical isolates, like the leishmania isolates, which you take from the patients itself. And I had both kinds of visceral leishmaniasis isolates and post-Kala Ajar dermal leishmaniasis isolates. So what you see here in the picture towards the left are the patients who have a visceral disease. Their liver and spleen are infected. There is a dermal manifestation of that disease, and that is called post-Kala Ajar dermal leishmaniasis, in which skin lesions happens. So I had both of them, and my objective was to see what is the mechanism of antimony drug resistance. And just to give you a basic idea, there are a lot of drugs used to treat leishmaniasis, and antimony is the first line of treatment. There are different drugs like amphotericin B, parmomycin, etc. And I had to screen all these clinical isolates. So I really need to have a high throughput screening assay, and that we didn't have. So my first goal was to develop that in vivo high throughput drug screening assay. And I was able successfully to accomplish that. And I could see that there was a good correlation between clinical outcome and IC50, like these are inhibitory concentration values for the drugs against antimony, and no cross resistance was observed, which means if the isolates were resistant to antimony, they were sensitive to other drugs, which are like second-line treatment, miltefosine or, parmomycin. So at least there is a hope for the patients that if they are resistant, they are not getting better through antimony, they may get better through the other drugs. When I established this assay, we got proposals from a lot of people, collaborators from outside in US to test their compounds. And one of the very promising compounds right now, and that is in clinical trial phase, is arylimidamides for the treatment of visceral leishmaniasis. And this is what we did as a collaborative project. Then the most important part was that, okay, well, now we have an assay. What is the molecular mechanism of this resistance? How is it happening? So just to give you a glimpse, there is a protein called AQP1. It's an aquaglyceroporine 1. As the name sounds, right? Aqua, it should take water, right? But it also takes antimony inside, like this is a water channel, and it allows the antimony to go in the parasites. And what I could see is that there was a downregulation of AQP1 in antimony-resistant isolates. That means these isolates are very smart, right? If they want to be resistant from the drug, they don't want to get the drug inside. And how would they do it? They would just downregulate, means the gene will be downregulated, it will less express, therefore the drug would not go inside. And that's how I could see that, well, this was interesting, that these isolates are different than the sensitive isolates, because sensitive isolates had upregulation of this gene. The resistant isolates were having downregulation. I was interested to see that what if Leishmania has, for example, other aquaporins or not, because plant has like 5 total aquaporins. And I wanted to see that, is this resistance mechanism only exclusive to AQP1 or other aquaporins playing an important role as well? So I did cloning and characterization of 4 other different aquaporins, and they were all similar to plant aquaporins. They were not involved in the resistance mechanism. The other genes which I could find for resistance were MRPA, ODC, and gamma GS. And the reason only I'm showing you this is how cool it is that, a parasite is so intelligent that they not only downregulate the pump which allows the drug to come in, but they also upregulate the pump which actually sequesters the drug. So even if the drug has come inside, what will happen? The MRPA, it is a multi-drug resistant pump, and it will sequester the drug into a vesicle, and that way the drug is not able to hit the target. So these parasites are very intelligent, right? And they have their mechanisms of resistance. So I did establish a very proteomics method for our lab for the first time to have the differential expression of proteins in antimony susceptible and resistant isolates of Leishmania. And during those times in 2008 or something, the proteomics was kind of very new. We didn't had in JNU itself the iTrack and SciLec methods established. So I was the one who established the method. Our professor was dean at that time. We could buy the machines, and with ICGB, I was establishing all those methods. Then there was another publication about, hydroxylamine esters to see the anti-leishmanial effect, just to give a glimpse. So when I did PhD, every day I thought, it's for me, right? Every day I'm spending in my lab is about myself. I have to do it to excel not only in the techniques, understand the science, but also to contribute in the knowledge of science, what field already has. And with that passion, I thought maybe since I understand leishmaniasis a bit, how about I understand cancer biology? And I moved as a postdoc in NIH, in the lab of Dr. Myung-Hee Park at NIDCR, and I wanted to understand the cellular function of polyamines and eukaryotic translation factor, EIF5A, for the prevention and treatment of cancer. So in your life, science is You should not be thinking, oh, well, I am an infectious disease expert. Now I cannot make a pivot to cancer biology. You can, the fundamental principles and the techniques remains the same, just the model system changes sometimes. So just to give you a brief idea, what are these polyamines? I know some of you already may be knowing that, but just to give you a brief idea, polyamines are putrescine, spermidine, and spermine. These are actually polycations. Like you see the calcium, magnesium, they has positive charge. They have a lot of positive charges. So they bind to RNA, DNA, and protein. It's just their nature, right? Polycationic nature. So they will bind to these things. So they are very important. They are present in every cell. And polyamine homeostasis is very important for the cell. Because as I said, if there is a disruption, there could be diseases, like there could be cancer, polyamine. So this whole homeostasis is maintained by a very good pathway. And I'm not going into the details of the pathway, but the pathway starts from ODC. So, ornithine. Ornithine, from ornithine, ODC works on it and converts into putrescine. So ODC is ornithine decarboxylase. And then there is a catabolic enzyme, SAT1. So what SAT1 does is spermidine and spermine. So it catabolizes spermidine and spermine into putrescine. So if the putrescine levels are low, ODC will make putrescine through ornithine to putrescine. If spermidine and spermine levels are high, SAT1 will kind of catabolize it. This is how the polyamine homeostasis is maintained in the cells. And I said that, optimum levels are super, super important for normal growth. If they high, transformation or enhanced growth, what that means is cancer, right? And if it gets low, there is an inhibition of translation, initiation, and apoptosis. And that is what I found in my postdoc. This was kind of we knew that polyamine does a lot of things, but what it does as a primary effect, exactly when it is depleted, it was unknown in the field. And from my research, there was the data came very clear and sound, suggesting that there is an inhibition of translation initiation. And I would go very briefly into that. But that being said, a lot of cell-based assay I learned new, which I didn't learn in my PhD at that time, like confocal microscopy, cell biology, and a lot of other biochemical assays. I had exposure of proteomics, but I did learn transcriptomics and epigenomics at that time. And that is very cool these days, ChIP-seq, RNA-seq, microarray. And that is, again, a very good field. And I can tell you, because my husband is a bioinformatician, he chose to pivot from wet lab research to just data analysis. And there is so much data with these high sequencing platforms these days that people are needed, especially the PhD trained people needed to analyze that data and make sense what is coming. Like, if this gene is coming up, what does that mean? If this gene is coming down, what does that mean? So there is a lot of requirement of bioinformaticians in current days as well. So few examples of my research findings, and the reason why I want to share it is because, the research is very serendipitous, and sometimes we actually, neglect some findings in the lab. And when I joined this lab in postdoc, there was very weird thing happening. I was told, okay, you have to overexpress SAT1 with the GFP, because they wanted to make a stable cell line. And I'm like, okay, well, I will clone the gene, I will overexpress it with GFP. And what I was seeing is that when I overexpress them together, GFP was inhibited. And I was like, why is it happening? Like, is the plasmid, when I put both plasmids together, are they not going into the cell? Or if they are going, why one of them is not making the protein? And I struggled with it for like 1 month. I did cloning different vectors, different systems, just to make sense of it. And then I found, let me do RNA fish. So what is RNA fish? And you may have read it in your books. It's like a fluorescence in situ hybridization. So what you do is that you label your mRNAs. And mRNAs, when they are labeled, you can see them in the microscope. So what you see here is, let me go through this very quickly, is the yellow thing, on the top left of you, GFP mRNA, right? So these are all GFP mRNA being synthesized, exported out of the nucleus, and they are making GFP protein, right? It's adenoviral GFP. There is no SAT1 mRNA, of course, because I haven't put SAT here. When I did SAT1 transduction, I saw that SAT1 mRNA is being made. When I did both of them together, what I see here is that GFP mRNA is being expressed, SAT mRNA is expressed, but there is no GFP protein. Isn't that cool? Why is it happening? Like, why there is no GFP protein here? So I was kind of, I was super, super surprised. And I said, I want to pursue that. This exactly, what's the mechanism here? What's happening here? And of course, SAT1 is a catabolic enzyme. So I knew that, polyamine levels would go down. So I said we had a, machine to do HPLC and measure these polyamines. So I said, I want to, prepare the lysate and I want to run these samples very quickly and see what exactly happens. And you see the putrescine is being accumulated in SAT1 overexpressed cells, right? That is what will happen, because spermidine and spermine will catabolize, and these putrescine and N1-acetyl spermidine, which are the byproducts, will become more. But what was the cool thing is, spermidine and spermine was getting depleted so drastically, like within 12 hours, there were undetectable levels of both of them. And when I showed this result to my boss, she said, this is awesome. This kind of depletion has not been established before. And she said, how? I said, what do you mean by that? She said, because polyamine depletion by other methods take long time. And it takes like maybe 3 or 4 days. And by that time, what happens is polyamine homeostasis happens. So cell starts overexpressing ODC, tries to compensate the polyamine depletion. But in this case, when you do this viral transduction, it is so quick that cells are depleting the polyamines very quickly. And within 12 hours, there are no detectable levels. So this could be a good system to see what is the exclusive role of polyamine. And I told my boss, is it possible that, the polyamine depletion is not just occurring the loss of GFP, but it is occurring the loss of any protein synthesis. In the cell, no protein is being synthesized, and that is what was happening. So I think you may have heard about it. There is a cool mechanism to do leucine incorporation. Like, there is a tritium leucine, radioactive leucine. Leucine is an amino acid, right? It will go to every protein. So you can measure the total protein synthesis by leucine incorporation. And I did that. And what I could see is that, okay, well, in these cells, there is a complete inhibition of translation within 24 hours. So that means spermidine and spermine both are essential for translation in the cells. At that time, I knew that. I wanted to go further and see whether it is translation initiation or elongation. Like, there are different steps of translation, and translation machinery is very, very, I would say, like a very good thing. It's a lot of initiation factors, elongation factors, and a lot of things. But the cool thing was that polyamines were required for translation initiation. And I did that polysome profiling, which, some of the investigators here would understand what does that mean. But just to throw that out here, we could see that the polyamines were required for translation initiation. But that being said, how would you convince the reviewers that it is polyamines? We knew that it is polyamines, right? So I had to do some rescue experiments. And there was an analog of spermine, that is benzspermine. So the beauty of benzspermine is that it is not catabolized by SAT1. So SAT1 doesn't recognize it as a substrate. So when I put benzspermine into the system, into the cells, what I could see is that as soon as I put there is no benzspermine. Of course, there is a translation inhibition. When I put benzspermine, you see the red bar goes up, which means there is a rescue of translation. that shows that the spermine is the one, which is contributing to the translation. Even if you don't understand anything from this slide, what is the take-home message is that if you find something very unique and it doesn't start making sense logically, pursue that. Think ahead and, think what could be the different ways. I remember brainstorming with different researchers. What can I do to find out that what's happening in my system? I didn't give up. When SAT1 and the GFP were co-overexpressing together and there was no GFP, I could have given up and said to my boss, well, you know what? This project is not working. I don't know what's happening here. Could you give me another project, please? No, I did pursue it, and it led to PNA's first author publication. And, this is the important part of it is not just about the publication. The discovery actually led other scientists in the field to follow this approach. And this paper is so well cited right now that people are pursuing this. That is the importance of the research. That if you do something, if it is solid, if it is, reproducible, people will pursue it. And they will use it in their systems. So I think I had a gratitude of that. Okay, well, my nights and days were well spent in the lab. The other project was eukaryotic translation factor 5A, understanding the cellular function of this. And again, this is a very cool protein. EIF5A is the only protein in this whole system which has a unique post-translational modification. And I'm pretty sure you may have heard a lot of different post-translational modifications, but this is called hypocyanation. And it is a two-step approach that hypocyanation happens. And this moiety actually, aminobutyl moiety comes from spermidine, and it is transferred by a DHS enzyme, which is deoxyhypocyan synthase, and then DOHH actually hydroxylated. It's the unique protein, right? It has a unique post-translational modification. So definitely it has implication in different diseases, cancer, diabetes, malaria, HIV-1 infection. DHS has also been identified as markers of neoplastic growth, metastasis. What does that mean? They are important in, disease systems. So it's important to find inhibitor for this cool protein. So we did knockout cell models, we did knockout mouse models, and we did global quantitative proteomics to deplete EIF5A and develop the system to deplete EIF5A and did the quantitative proteomics to see what happens in the cell. That was published in scientific reports. And then I wanted to pursue further to actually find inhibitors of it. And we did high throughput screening of chemical libraries, more than 200,000 compounds. And now you must be wondering, wow, that's too much. How you must be doing this 200,000 compounds. So there is a robotic system, at one of the institutes at NIH. We had developed a collaboration with them and said, well, these are the libraries. Now your robot can do it. You come up with say 100 molecules and we will test in our lab. At that point, when I was talking to different entrepreneurs and investigators about these things, like how the drug development happens. And this is my next slide, but I will take a pause here because it was around my postdoc. I was about to finish my postdoc and I was thinking what exactly I want to become. And I got deeper and deeper interested into the implication of science, into how the drugs are developed, how actually what is the clinical development. Whatever we do on a basic science research, it is one thing, but there is, of course, a part of clinical science. And the dysfunctional belief could be that, oh, well, now I am a postdoc, I have to find a academic job to pursue my ideas. I'm stuck here, because nobody is going to give me funding to test my inhibitors. There is a lot of money goes into drug development, millions and millions of dollars. So, but then I thought, wait a minute, I'm never stuck because I can always generate a lot of ideas and I can always help people who has ideas. So if somebody has a drug development in their lab, I can help them. as a clinical scientist, as a somebody who can and who understand the science, who can write the documents and blah blah blah. So I thought maybe I just have one path to go. I have maybe one right idea that I have to be a clinical scientist now in one company. But I thought there are again number of possibilities here. I don't want to just work on one molecule whole my life. I want to learn different things. So what I can be. And this was the time when I was brainstorming with a lot of people, having informational interviews. I was doing my research in the lab, but then I was talking to people. I was writing to people like what exactly I can become after doing PhD and postdoc. And I'm so passionate about clinical development. And then I took a lot of courses here. NIH is a great place, but I think everywhere you get a very good exposure to drug development. What are the different steps? What kind of trials happens? And I did those courses, met a lot of people from FDA, and learned about these clinical developments process actually. And I would just briefly go here to give you an idea that, the pre-discovery, the preclinical development of a drug takes 3 to 6 years. As you see in this blue zone, right? There is a drug discovery, 5,000 to 10,000 compounds. Initially you screen. Then they goes to preclinical. Preclinical means you're doing it on the cell lines, you're doing it on animal models. FDA requires at least 2 animal models. So you're doing all those kind of assays, and it takes solid 3 to 6 years. Then there is an investigational new drug application submitted to the FDA saying that, well, we believe that this drug works very good in these 2 animal models, in these cell lines, and we want to test this drug in humans now. So before it goes into human, FDA is an authority in US. Similarly, there are regulatory authorities in every country who approves the drug applications. They very carefully look at your proposal, look at the objectives, endpoints, what exactly you want to achieve, because you're going to inject this drug into human route. And now everybody after COVID, I'm pretty sure we are very familiar about clinical trials, right? Everybody is getting this vaccine got developed, through clinical trials. So there are different clinical trials, phase 1, phase 2, phase 3. Any study starts with phase 1, safety and tolerability. Want to see that whether this drug is safe or not. Then phase 2 trials happen when the number of volunteers increase, like from 20 to 80 to 100 to 300. And then phase 3 happens where 1,000, 3,000. And right now, as you can see with the vaccine, lots and lots, many, I don't know how many thousands of volunteers. And then NDA gets submitted and then FDA approves a drug. So it's a very long period of time, right? It takes at least 10 to 15 years to get a drug developed and get to the market. So now what scientists did, and I just want to make a pivot here because I want to emphasize the beauty and the power of science. In COVID pandemic, you imagine so many vaccines, like at least 3 of them got developed within an year. So isn't that extraordinary? Isn't that like how much work they have put in just 1 year to get all of this process? Of course, this is a drug development. The vaccine development happens differently, but it's still, the idea is same. They have to go through all these things, all these regulatory steps, trials. So people really worked hard. And I wanted to work somewhere in between the green zone. I knew that that's where my heart is. I wanted to see from my PhD itself that what happens to this drug to the patient. And I was working with the clinicians and then getting those isolates at that time. And bottom in my heart, somewhere I wanted to be a real medical doctor, to be very true with you. I didn't become that. I couldn't clear actually the medical exam in India. So that was the thing. It was always in my heart that I want to be a doctor. I want to work with the doctors. And I thought maybe this is the time, I can work with medical doctors who has cool drugs and they are working on cancer therapies. And I joined NCI as a regulatory medical writer. Okay. So NCI has Center for Cancer Research. It's a kind of 250 different labs working on cool things like therapeutic cancer vaccines, immunotherapies, and NIH has the biggest hospital. So we have a research hospital where you can write the clinical protocols, develop these therapies, and give it to the patients, because those patients has exhausted all the different treatments available in the drug. So they come for a hope to the NIH, like, do you have something for us? We have melanoma, we have gastric cancer, or different kinds of like head and neck cancer. Do you have some hope for us? And I work with those investigators to write those phase 1-2 trials. So this was my heart of regulatory writing, and that's how I become a regulatory writer. But I want to give you an overview. Not everybody is interested in clinical trials, right? So medical communications is a very big umbrella. And you can do different things. You can do editing, you can write educational materials, you can write informational materials, you can write promotional writing, like, promoting a drug or promoting a therapy, promoting a compound. Publication writing is one of that, because when these phase 1, 2, 3 trials or any science is published, right, people need writers to help them, and that speeds up the process. In US, they always have some in-house writers, or just helping them so that scientists can focus on their science. Writers can write the publication sometimes if they, so the bigger groups, like for example, my group I work with, they have a dedicated publication writer. Then there is a scientific non-clinical writing. There is a medical affairs, medical liaisons. That's again a career. So I just wanted to tell you here that you have to find a niche here, exactly what you want to do and where your heart is, where your passion is, and go for it. There is, like, if you like writing, if you are someone who likes to project their ideas, start it. At very early age, you can find a lot of volunteer opportunities to write. So, okay, so this was in my head, this was in my heart, but how would I convey it? Not only to myself, the inner, deeper myself, to a scientist, right? Because once a scientist, always a scientist. And am I leaving the science? That was the first question I had. Would this transition mean that I would gonna miss the lab? I'm gonna miss my experiments, my individual contributions, my freedom? No. So that was one thing which I had to explain it to myself. Not because before you explain to others your passion, you have to first convince yourself with that idea. And I thought hard that no, it's not going to be. The transition to medical writing is not a leaving science. I am going to enjoy more science because I'm going to learn different kinds of science. Here in my postdoc, I'm just doing like polyamine research or EIF5A. But when I will be working with different investigators, I will be learning about their science. I'll be passionate about what they are doing. That was one thing. The second thing was I like working with data a lot, but not necessarily collecting it. I'm not, I was not passionate to develop the Western blots in the dark room, but I was passionate in looking the blots and like what exactly that means. And that's what I am doing right now, right? When I have all these pictures and data, I have to write a summary about it and what does that data means. I was very, the good thing is I'm very detail oriented. That is the one thing, like, I knew from beginning that I'm down to the number of spaces used in the document. Oh, there are 2 more spaces here, it should be cut to 1. So that kind of detail orientation, just an example, but you need to have a very, very detail oriented mindset in the regulatory writing, because these documents are super, super important for the drug approval, right? And they, whatever we give to the patients and whatever we submit to the FDA has to be absolutely accurate. Absolutely. There is no chance of any error there. So that was one thing. Then I'm a process-driven person. I knew that, okay, well, I love to follow SOPs. I love to read regulations. I love to do exactly like, this is the rule. We are going by this way kind of person. I'm a forward-thinking person. Okay, if we have, for example, IND application waiting, what we can do with the IRB stuff, like institutional review board, like, planning kind of thing, forward thinking. Okay, if we have 1 month of waiting time, what else can we do with this project? And planning everything ahead. I wanted to write and not just get a job outside of the lab. That is the most important thing as well, because when I was trying to switch, it was important that I stick to it for a long time. It should not be like I'm moving after one after the other. So I spent my time, and I did a lot of informational interviews to make sure what actually I like. Because as Manju has told, that I did a technology transfer internship. So I did work on technology transfer agreements. I did work on patents and marketing abstracts, but I didn't enjoy that much because I thought that is more of a legal and complexity. But I wanted to do the science, a clinical science. My heart was with the patients. So I didn't enjoy much of the law part. So knowing what you want to do is important, but knowing what you don't want to do is more important, because then you will clear out your path. Okay, this is what I want to do, but these things I don't want to do. I was a flexible, and like always, I'm a flexible and team player. I knew that from in the lab itself, like we both of us, Manju and I, did a lot of, experiments together. And even in my postdoc career, I was always a team player. Let's do it together kind of thing. And I knew that regulatory writing is going to be a team adventure. It is not a solo writing. Lots and lots of people involved in one document. And I enjoy it. I really, really like it. So you now must be wondering, okay, well, now you are a writer. You must be having a computer, a screen, and writing, typing. No, that's not the day look like, okay? You are a writer, but you are also a manager of that project. So lot of thing happens in a day of a medical writer. There is, of course, a writing thing, scientific, non-scientific, and you need to have a technology and software proficiencies, like just not Microsoft Word, but a lot of different systems. We have to submit the documents into certain systems for FDA, certain systems for approval to the IRB. So those kind of software proficiencies are needed, and you learn it. That's not a big deal. Document management is another part. There are so many version controls, because it's going through different review process each time, scientific reviews, FDA reviews, institutional review board reviews, lot of reviewers' comments comes and goes. So you have to have a very good management of documents, how you're going to save them and keep a track of them. Then there is a lot of communication, right? Investigators' communications with them, study sponsors, monitors, IRB members, a lot of emails, phones, teleconferences, because it's not just my decision to make I'm just giving you an example that, okay, if there is an exclusion criteria or inclusion criteria for a trial, it has to be debated into the team itself. And we are enrolling this patient, what we exactly want to have. So everything has to be everybody has to agree with it. So a lot of communication. And if you are good into that, so go for it. Leadership is like pushing forward a project, running a meeting timelines. If FDA comes for review responses, they give us sometimes just 24 hours, 48 hours. Maximum is 72 hours. So you have to get things done and submit in that time. So in that time, you have to have a team together and get things done. Especially the timelines get very shorter for IND submissions because those are small study submissions. For NDAs, they give more time. But even though those are big documents, can you imagine INDs are 200, 250 pages documents. Project management is the most important part. So if you get a chance to do some project management certification or understanding the project management, what it means. You have to juggle with different concurrent projects in different indications, managing many moving parts, acquiring documents and data from others. So you have to be very good in project management. And I say that project management is crucial for any job. Even PhD itself is a project management, because in PhD, you're not going to have just one project, right? You're going to have multiple projects, and then you will be working with others on a collaborative projects. So you have to have a good understanding of how you're going to do multitasking, how you're going to manage your day, get things done at the end of the day. So I think that is a very important skill to learn from the beginning. Serving as a resource for team, you have to be an expert on issues related to protocol development, monitoring, as well as regulatory issues, because in the team, there are so many different people, right? They may not be aware about regulatory issues. You have to train them, tell them, oh, this is a new guidance. We have to follow this way now. Training is continuous training. Like, it's a continuous training. You'd have to keep updating yourself. And that is the thing I like, because no single day is boring. There is always something to learn new and teach others, contribute a lot. So I feel personally that I'm very lucky. I was very passionate to do this kind of job, and I got into this field. But somebody who is trying to understand the clinical development, or maybe you don't want to be a writer, but you want to be some clinical scientist or a medical liaison, I'll have these helpful training and resources, which will be very helpful for you. Some of them are free, some of them have a little fees, and you can start, with your master's. There is never a kind of like a threshold that, okay, well, you should be a finished master's to learn something new. You can learn something new anytime in your career, right? And always make sure, and that is something I learned when I came to US. Besides your projects and work you do, there should be always a 10%, 20% of your time in self-learning. You have to spend that 10 to 20% to grow into the career itself. And that is the time you have to find. My days get very hectic, but I try to find some time when the days are less busy to read something new, or to attend a webinar, to attend a course, which I can. So here you go. So for example, I did advanced studies in technology transfer from FAES. This is a Foundation of Advanced Education in Science at NIH, and they had like 2 years of program. They had a lot of different courses about biomedical business development, patenting. There was FDA regulations. So it was a very good, strict courses, but I'm pretty sure IGNU has one because I looked into that. They have similar courses as well, teaching you how this works. Duke's Office of Regulatory Affairs and Quality Regulatory Affairs Training Program. It is a 6-week program online that introduces you to the clinical development process and where documents fall along the drug development timeline. And you don't need to be just a writer to understand, and you can be a clinical trials coordinator, right? There is a huge demand of clinical trial coordinators because they're in every country on this earth. Clinical trials are going on all the time, and people are needed who can actually coordinate those clinical trials. So you need to have an understanding of these clinical trials. EMWA resources. This is an American Medical Association for Writers, and this is a great resource. Like, they have fee-based some of the certifications which you can take, and they also have freelance opportunities, active discussion forums, and there is a very discounted fees for students. Introduction to the Principles and Practices of Clinical Research through the NIH. This course is open annually, but the previous lectures are available on YouTube. Very good course. I would recommend it because it teaches you the ethical aspects of clinical research, what you need to take care of, when you write those kind of documents. Be patient-centric. Think from the patient's perspective as well. Regulatory Affairs Certificate is one of them, RAC, and people in India do a lot as well. Strong credential in regulatory affairs, but I would say that you need an experience to do that kind of course. At least 2-3 years of experience. Drug Information Association Clinical Research Certificate. This is a quick way to get a certificate that indicates an understanding of clinical trials. Then there are, of course, university courses and master's degree, but you are already doing the master's. So there are more, like in some of the universities here in US, they have like a master's in regulatory affairs. But I'm pretty sure what you are doing right now can be used as your understanding. You can always build that regulatory understanding from all the different resources here. You don't need to do a special master for that. Read FDA guidance for industry and EMA guideline documents. Those are kind of free as well. FDA guidance, all free. Okay, you just have to have a Google. Medical Writing Competency Model. This is a very good publication I quoted here, and this has all the competency things, what you need to have to be successful in a medical writer. And that's free, so you can have a look into that. So with that, I think I would pause, and I would thank everyone for listening me. I don't know how long I spoke, but thank you so much. And I would invite any questions you have. And I just wanted to say that career is, for yourself, it's a long journey. And though I will be finishing my 10 years at NIH and 3 years, I am looking forward for more opportunities right now. And I am going to start private sector very soon this month. And I will be a senior medical writing scientist at Johnson & Johnson. So there is always a growth, and there is a bright future for you all, guys. I wish you best of luck. Thank you.
Thank you, Dr. Swati. This is Dr. Ravindresh. I'm a colleague of Dr. Manju's at Central University of Punjab. First of all, congratulations on having a wonderful career so far. It's amazing. And the best thing about your talk was that it was inspirational, motivational, and I think as Indians, we could all relate to how your career journey has been. For example, we have all, done our PhDs here in India, then went for postdocs outside. But you have definitely taken the road less traveled by, and I guess that has made all the difference in your career. Also, I would like to thank you for doing this at such an odd hour. I mean, waking up so early and doing this for us. So we really appreciate you talking to us. Maybe in future we can, have you here physically present with us at Central University of Punjab. So there are many things that I really liked about your presentation, be it your PhD work, which you spoke of, working on leishmaniasis, and then moving on to cancer biology. And what is remarkable for me is that you excelled in both the areas. You have excellent publications, and also your, that experiment where you talked about the GFP not getting expressed in the presence of SAT. I think that was really exciting, and that is something that I've seen in a lot of researchers, because many a times we kind of, leave that out and we don't really work on things which are exceptions. And I think this is very important for the students who are listening to this talk, is that in science, you cannot, go about expecting things that, okay, this is the only way things can happen.
That's true.
You have to be ready for, you have to be ready for the new things that are going to evolve. And that is how you make a difference in research. Because if I know that, this is going to be the result and this is all I expect, I will never look into the unknown. So that was very exciting for me. And then you're talking about the passion, hunger, and drive. I think that's a wonderful way to describe PhD. And many a times, these students ask us that whether we should do PhD because we are really not interested in doing the bench work. So you, I think, summed it up very nicely in your presentation, that why you need to do PhD. PhD is not just doing research, it's also building up your personality. So that was interesting for me. And I think another takeaway from your talk for all the students would be that you should never stop learning. Like, you were a postdoc when you started doing all these amazing courses. And I think that is something that all of us have to learn, that we have to continuously be the students of science, different aspects. You cannot restrict yourself to just one area or one thing. That's not how you grow. So that was also pretty motivating, I think, for me also, that we have to continuously keep evolving, keep learning new things. So that was also exciting. So with that, I think we can start taking a few questions. So let's see what the students have asked.
Thank you so much,
So,
Ravindra. I think this was, I would just take a minute to say, I think you very well summarized all the things which students should take as a take home message. And we are all students, by inherently, inherently somewhere in our heart, we crave for learning. We want to grow and learn more and more. And at very young age, as you think, that your career can grow exponentially if you take it seriously from the beginning. And it was very good. And I think students are lucky to have you and Manju and Dr. Ramakrishna as, your professors who are exposing them to these kind of options early in their career. Because at my time, we never talked about these things. For us, it was like kind of a tunnel vision. We have to do our masters, do PhD, then become a academic, professor somewhere. And I learned this when I came to this country, right? Because the students were very much exposed to all these different options they had. So it's wonderful to have this kind of series as well. Thank you.
Yeah, I think Dr. Manji needs to be thanked for this initiative. I mean, she is starting very pertinent series for all the students. Many of the students are actually at crossroads right now, and they are thinking about what to do next. And many a times they're stuck, because I've seen that many of people, they leave out of science in India after doing PhDs and after even doing masters, and people who
That's
are
good.
rank holders in universities, they're leaving out of science. So I
Yes.
think Dr. Manju needs to be acknowledged here, that she has started a very pertinent series which is going to be useful for all the students.
Yeah, I would congratulate for that. And also, especially in women, I don't want to make a distinction here, but, women after marriage and after having kids, I have seen a trend that they drop out, even though they have had an exceptional PhD career or, postdoctoral careers. And for those women, I would say that the road doesn't end there. There are so many different options. You can work from home, you can have the work-life balance and get things done. So
Yeah. Right. So with that, I think we'll take some of the questions that students have asked. So Abhijit Naik says that, does the number of years of PhD matter? As in, how long is your PhD? Does that really matter?
So I would say no. The quality of the PhD matters, the quantity not. So for example, I would be amazed to see if somebody in biology research will finish PhD in 3 years. I think 5 years is something. I personally think 4 to 5 years is the time when you kind of get things done. If you feel that, oh, well, I am just in second year of my PhD, third year of my PhD, can I submit it? I am not too sure about that. It's PhD, like you will start with a project, you would have a synopsis. You have to find something novel in the field, which has not been established before. And in not talking about physics and chemistry here, but especially in biology, it takes time because things doesn't move as smooth as we wish. Experiments doesn't work in one go, and you need a reproducibility. Even if you got a Western blot in one shot and you think that's cool, you have to do it 3 times to make sure that what you got is correct, right? And then publish it. Publication itself takes time these days. I see a trend that these days there is an extensive review process. So 3 to 6 months goes very easily into a publication. So I would say have a reality check. For me, 5 years was the deal. I was like, I'm having 5 projects in my hand. I would do all of them good. And 5 years is the one I want to finish my PhD. I know my supervisor wanted to have me for longer. And she actually, we had quite a heated debate towards the end of PhD. She was like, you hold here for a few more years, 2 or more. We will finish. We have 7 papers. We will have 3 more papers in one next year. And I'm like, I can teach other students. I don't care if I will have them as a first author or not, but I'm going to train others. And that's all I can do. But then it's their journey. they should take it, they should publish it as a first author. So I was very clear about it. But if you feel and your supervisor feel that, you know what, if I will leave, nobody else is there in the lab to finish this experiment, then, maybe you have to stay. So it depends upon the circumstances. For me, it was a big lab and people were there who could take my projects moving forward.
Yeah, I kind of agree with you, Dr. Swati Dadhich, that you have to negotiate with your supervisor sometimes. And like you said, in biology, 5 years is like an optimum time because there are so many techniques that you need to learn. It's a training process. So you shouldn't always rush through, but at the same time, you should not really extend it beyond 5 years also. The ideal tenure for a PhD would be 5 years. Yeah, right. So Mohabbat Singh has a question. Your tips for aspiring science writer?
I would say write as much as you can, because writing is a skill that you learn as you do more. I would not say like, okay, I'm an excellent writer now because I am a writer. No, it's a practice. And take every venue, like every opportunity. For example, if in your university there is a newsletter, volunteer for it. Say, I want to write this section. It's a short story. You want to write something. Or like these days, a lot of science communications are about making cool, designs and explaining a layman people what the vaccine is. For example, these days, telling them that taking vaccine is safe. You can write in the newspaper articles about it, and so write as much as you can and take it seriously. It's not a copy-paste business, okay? Don't do that at all. You can take ideas, you can read as much as you can from other people's, other publications, have the ideas, have them somewhere, but then put your thoughts in. And then go back and cut the dead wood. It's all about finishing cutting the dead wood. Like, okay, your first draft is not going to be the excellent, but cutting that and then making it another draft, looking back it again and again. And I would encourage that whatever opportunities you get around, seek for it, write as much as you can. Even on the social platforms, like I put a lot of LinkedIn posts and I like, okay, this is a new discovery. How would I summarize it in a paragraph? And I would write like few lines about it. And somebody told me, and that was very interesting, when you understand things better, then you explain them better. If you have a poor understanding of something itself, you won't be able to translate it to your audience. You have to build an understanding and then write it and recognize your audience. If it is going to be a layman, people like, for example, when I used to go to Jaipur and my parents don't know anything about research, and I would tell them, that was my job, okay, what I am doing, what is Leishmania research? I'm going to explain you. And I was very even in the trains and buses while I'm traveling, people are like, oh, well, somebody used to say, you're doing PhD, do you have any job? And I'm like, no, PhD is my full-time job. I'm going to explain you what PhD looks like. So do write as much and do read.
So I can relate to that explanation that you have given, Dr. Dadhich. So maybe you can, you on full screen. And
Sure.
I used to do that with my grandfather. My grandfather used to ask me that you're not a doctor. So what exactly are you doing? Are you going to be making medicines for all of us? So, it was a great experience for me to actually explain to a layman like my grandfather, who had come from Pakistan, who was not that educated, but he was interested in what I was doing. And that kind of makes it easier for me now to explain my science to a layman. So, Dr. Jan, would you like to take a few questions? Your mic is on mute.
Yes, Dr. Dadhich, right in the early morning hours, I see you are full of energy and enthusiasm, and I'm assuming most of the students are really, they are just enjoying and stuck up with the talk, right? So I see that this, transition, so-called transition, I would say, it's basically not a transition for you. I can very well relate to you. And actually, as I was also in my, during PhD, I was into Leishmania research, and I used to have those WHO sites and all. And I used to wonder, oh, sometime I'll get to go in this WHO thing and actually work in the field and, make things implicable at that level. So, and right now I'm suggesting those things to my students actually.
That's
that is one option. You can go ahead with that, right?
Yep.
So because one cannot do all the things in one lifetime. We can always try. As I see you, your spirit is like it's one life and try to do whatever you want to, right?
Yes.
Yeah. So here I would like to put a question from my side. So do you think the intricacies of biological experiments which you learned in a laboratory environment, be it master's or PhD, has helped you later in terms of documenting work, like those intricacies?
Oh yeah, definitely. So when I talk about these clinical trial protocol documents, right, they do have a very good background section of like 15-20 pages, and investigators give me all those kind of next gen seed data, sometimes the cell toxicity data. I have done those, right? That's why I understand them. What does that mean? And narrate them exactly how that experiment might have worked. And sometimes I ask them, can you provide me a better picture? And because this picture is not good, it doesn't look to me to be in a regulatory document. And they say, well, just to hold on, my student is finishing his third, kind of finishing up to get it a better image, and I will give it to you. So you cannot get that kind of understanding if you haven't done it yourself. And I see that kind of difference and I'm not saying that people who are not a PhD and medical writer are not good ones. They must be with their experience, right? Because they have accumulated a lot of experience in the field itself. But when you have a PhD, you have an added advantage. You know those things already. And it is not hard for me to discuss the data with them because I understand it. And it just gets easy for them as well. They get very comfortable and they value it. They take they consider me as a valuable contributor rather than a writer who is just ghost writing, like, okay, in the background, just putting the data, because they know I can question them. They know that I would ask them, for example, if I'm putting the data, I need to make sure that it is referenceable. There is a reference for this data. And most of the time, if it is unpublished, I would say I need a reference when it is published. And sometimes I would go into the reference itself, go better seek the supplement data just to make sure that it is in sync with all the document, it's consistent. So yes.
So coming back to another question, like on a personal note, I would say, like cancer biology. I mean, there's a big community of researchers, the basic biologists into this field, right? And you won't believe, most of the PhD aspirants which are coming to Central University of Punjab, if we ask, what is the kind of work you would like to do? And the first thing, and almost maybe 90% of the cases, it is like, no, we want to do cancer biology, right? So definitely, because maybe this is one of the most commonly heard around problems. But coming to your experience of being a basic biologist in the field of cancer in your postdoc, and also the approval, the clinical trial stuff again in the same area. So, what do you think this basic biological research going on in the laboratory, how it is relating to the clinical picture, the real clinical picture? Where are we? I mean, would you like to comment on that?
I would say that without the basic science, no clinical research can happen. Let's put it this way. If you saw these 3 vaccines coming in just 1 year period of time, there was 10 and 20 years spent in basic lab to establish those vectors, to establish the methods, whether that's going to work or not. So I know personally because, for example, the Moderna's vaccine was, all the preclinical research was done in NIH. In one of the institute, Dr. Fauci lead for NIAID, right, infectious disease. And they had a very solid preclinical data, the clinical data also. from the beginning that it's going to work, and that's how they could make it on a larger scale. So I would say basic science is the most important thing. You have to get it right from the beginning. The clinical research comes later. If you are always interested, you can always transition. And I would not say to box yourself into being a cancer biologist. Because you know what? Nowadays, you see, our life is stopped because of what? A virus. So what do you see? Like in 2 years, 3 years, the whole hype would be towards infectious disease, because people will say, oh my God, the world is stopped because of a virus now. So let's work on the virus. But you know what? The machinery, the basic biology machinery, the methods, the understanding of science is same, irrespective of what system you are working on. If it is like a kind of a parasite system, or if it is a mammalian, of course there are different genes, different pathways, but you would very easily relate to it. It's not a big deal
like in say for anti-cancer drugs and all. So we know there are different approaches: the rationalized and the the randomized. So any comment on the randomized one? Because definitely a whole chunk of publications are churning out.
Oh yeah.
On the screening component. So you think we are in the right direction, or I mean, as a person who has experience on both sides of the coin?
I don't know. I can't say. I have not been in India for like long time, so I don't follow their publications, right? So
I mean, international scenario, I would say it's not only like
so internationally, I feel that we are in a very golden period for cancer biology. And that's my feeling is because you see right now the cancer biology field is going towards immunotherapy, cancer immunotherapy.
Yeah.
After James Allison research for ipilimumab, right, which works on melanoma 10 years back, and they got the FDA approval, the shift is tremendous to understand how immunology works. So the one thing which I feel is the most important subject one should understand and do is immunology, because immunology is something is implicable in every disease, right? So if you understand that how T cells work, how B cells work, what are the dendritic cells, and those kind of concepts you have very clear, you can. And so I feel personally like to just answer your question, cancer biology, the next 10 years are going to be the golden period for cancer, because we're going to have a lot of drug approvals in next 10 years.
Okay. So let us get back to some students' questions. So here is Nikhil. So he's asking, how difficult will it be to get a faculty position in the top institutions of India in pure biological sciences?
Let's not, okay, how difficult it is going to be. For everybody, the parameter or the threshold of difficulty is different, right? So if I say it's difficult for me, the threshold could be different than what you consider it to be difficult. So I would just rephrase this question saying that, is it possible to be a biological, do basic science and get a position in a good institute? Yes, of course, there is, possibility of getting a science. But, that being said, your basic science research should be somewhere having an objective of implementation. Because why the government should pay money, right? And that's the tax dollar money, right? So if you are saying that I want to achieve this, there should be an objective saying why you want to achieve this. So, I always go with this concept: why in the middle, how, and then what as an outer circle. So I always go from inward to upward. Why we are doing this, how we will do is the second step, and what we will do is the third thing. So if you have your why very clear, whether it's a basic science or applied science, you are good. You will find out how you will do it. You will find out what needs to be done. But your why should be there. I don't know if I answered your question, but I think I kind of I would not say that it's super difficult to get in a basic science, then in applied science. Maybe it is. But if you have a solid basic science concepts that can be applied into different things for applied science, maybe you have a shot there.
Yeah, I think what you said is quite right in the sense that, you have to have a strong why and you have to be passionate about what you're doing. Then you will not feel the difficulty of it.
Exactly.
So if you're passionate, then you have to.
So, have you seen the James Allison? I'm sorry to just to cut you off, but I would say encourage people to listen the documentary for James Allison. And the EP story came from all his basic research, right? How passionate he was. And for so many years, people didn't provide him funding. People just cut him off, said, you know what you are saying about the PDL1 inhibitor, it doesn't make sense. CTLA inhibitor doesn't make sense. But he pursued it no matter what. He made his even personal sacrifices for that. And he went for that. And he got a Nobel Prize, right? So the passion should be like, you have to be very passionate about what you want to do.
Yeah, this story also reminded me of Shinya Yamanaka, the guy who discovered iPSCs for the first time. So
yes.
I attended a lecture by him, and he also mentioned the same thing, that people were calling him crazy for trying out this thing. So
I know.
You have to be passionate, and you have to be probably a little crazy in the eyes of others to make a mark in science. So, Dr. Dadhich, there's one question that I think for students I would like to ask is that, for example, let's say that somebody is doing their master's or let's say PhD in India and they want to become a medical writer. So can you suggest something that they should pursue here in India, or do you think that there are more options outside the country only if they want to get into the medical writing field?
I think there are a lot of options for writing in India, because what I see a trend, and I would see more when I join Johnson & Johnson. I know that a lot of outsourcing happens for Indian writers. So there are a lot of companies who work globally, and they get a lot of documents done, especially for clinical trials. Because clinical trials is just not about writing the protocols. There are so many documents needs to be written, so many summaries needs to be written, so much data needs to be compiled, and we need a lot of people for that. So there is what I could hear was that there is a lot of people back in India, and especially Indians, I would say, because we have an added advantage of having English, better than other Asian countries, right? And we are cheap to hire. Let me put it this way. So the companies go for it. So you have a lot of opportunities in India as a writer. And I think what you should be doing, if I would be you, go on LinkedIn, find people who are successful at writing positions in India. They are all working, because this job doesn't take you to be physically located. You can do it from anywhere, right? So if I need to get the document written, I will send it to you from here, and back in India, you will be working on it, send it to my time. So, you see, sun never sets. There used to be a kind of this thing for Britishers, right? That sun never sets for them. So the same thing companies adopt. They work the whole day, get it done overseas, then come back again next morning, work on it. So a lot of things gets done.
So do you think even after master's students should get into this, or do you think PhD should be a must?
It's up to you. Like, I see a lot of people going after masters also. What I see in regulatory writing, though, is that people who have PhDs, it's just a growing trend now. It's not that established trend. People recognize the value of PhD. And I would say if you have some reasons not to do PhD, like, for example, some family reasons or some financial reasons that you cannot afford to have PhD, go and have an experience after master's. When you feel like yourself that, having a degree would be beneficial for you, come back to it. The way I thought about it was that when I was young, like I'm young too right now, but, when I was doing my master's, I thought, let me do it in stretch right now. Because doing PhD after having a family, kids, it will be really challenging. And I knew that from other person's experience who were in the lab from Nepal, and he was sacrificing on his family time with kids and spending time in the lab. I didn't want it to do that. I'm like, I would just get it done out of the way and start after that. But, everybody's journey is different. Everybody is different and their situations could be different.
And do you miss bench work research?
Dr. Ravindresh?
Yeah.
Yeah, so I think we can, because I know Swati committed, say, for half an hour or so, because she's actually in between leaving and picking up the new thing, she's in real transition.
Yeah.
But I really appreciate the kind of time, and she's all engrossed, in her.
I know.
I mean, I see both of you just enjoying the talk, and, that is the whole purpose, actually. So, Aju, there's another student, maybe it can be the last question, Dr. Ravindresh, Dr. Swati.
Yeah, that will be fine.
Yeah, and so he's asking, what was your strategy during master's program to balance yourself with academics and acquiring knowledge about the research, the part?
Oh, sure. So first I will answer Ravindresh's question. He asked, do I miss the lab work? No, I don't. I don't miss that.
That's great to know. That's great.
Actually, I don't. And coming back to Aju, your question, when I was a master's, I had a plan that if I want to do a PhD, I have to get NET cleared. And you must be aware about this exam, right? NET fellowship, which is like UGC CSIR fellowship. There are tons of different fellowships these days. I wanted to have secure funding for my research. So besides the master's, program and the courses, I used to study myself for NET exam. And when experiments are done, I know that people were, masters you are in the college, having good time in canteen, having, good time, movie time, and, who does the experiments? And, but I was good into that. And as you may have seen, I was the first ranker, right? And because I was completing their experiments too, right? They were enjoying outside. And it is good. Sometimes it's okay to have fun, but if you have a clear objective in your mind, maybe you will enjoy doing experiments too. And when you will get better into that, you will enjoy them better. It's like that, when you get better into something, like, okay, if I can do this gel next time better, I will do it. So this, and I was always interested in teaching. So, for example, the when professor used to say, well, I am going, you can carry on the experiments. And somebody who's coming after a canteen break would come, oh, what did he tell? And I'm going to show them, okay, I'm going to show you what he told when he was leaving. So you have to take that proactive approach sometimes, being a leader. Okay, I'm going to show you, I'm going to teach you how it works. And it was an enjoyable experience for me. We have a lot of time in masters, to be very frank with you. It gets busier when you do PhD. So enjoy that time, have fun, but do hard work as well.
Yeah. So, Swati, at the conclusion, I would request you to say something about the STEM thing, right? How you are sort of manucuring through it and trying to help the upcoming science aspirants.
So I would say for STEM mentoring, what I do here is the Women to Women mentorship program. They have a women to women mentorship.org, and I'm passionate about it because I feel that sometimes, and it's not about making a difference here, but women needs to be encouraged to in the STEM programs because of the other reasons they feel left out. So what I do is I provide them 101 coaching, but that is in US perspective. You can always find some of those mentoring programs, and I haven't searched myself. I do know the links, some links for US where you can find mentoring. Like, for example, right now, what I'm training is a person who is doing masters in biotechnology. And I'm teaching that person what is a regulatory profession looks like, how she should build her career. And, a lot of personality stuff also, building confidence, how to approach interviews, all those kind of things. But I would say in Indian perspective, like for example, you are in University of Punjab, look for the virtual opportunities, whatever university have for mentoring, and if they are open for you. Do informational interviews. Go over LinkedIn, post a message, say like, hey, can I have your mind, like pick your mind for like 5 to 10 minutes? I would speak with you. And people are generous with their time. They will actually go ahead and talk to you about it. I have spoken to many people in my career like that, whom I didn't knew. I just connected through LinkedIn, and I just spent 5 to 10 minutes talking to them, got some ideas, and that's a mentorship. Mentorship is not just like having somebody, sitting on you, checking on you every day. You have to have your own circle of mentorship. Somebody would be like in your inner circle whom you will reach out on maybe monthly basis or like weekly basis, but somebody would be in outer circle whom you will just reach out once or twice in a year. And that is also considered as a mentorship. Did I answer your question, Aju?
Yes. So, Dr. Swati, really, I mean, you are keeping us just on, and we are really enjoying, your company and the things the students specifically. Although, honestly speaking, I was expecting a better turnover of the students being a Saturday, and although at the starting they were around 60, they are 50. See, so that is the recklessness, right? Not from the speaker's side, it's from the listener's side. Anyways,
okay.
Yeah, so the other thing, so I really look forward for both associations and interactions with you. And definitely the students can get back to her for whatever, questions they have still, like, in their box. So another thing I would like to mention, Dr. Ravindresh, maybe you can put up the slide. The second speaker of the talk has joined us. So Dr. Rupali, you can just please switch on your cameras if you can. It's possible.
Oh, I am not in the situation of switching on my camera.
It's fine. It is just students and Dr. Swati. In fact, last night I was talking with Swati, and she was really like, I was showing her this thing. Okay, I'll just flash it. And she was like, oh, I really want to get in touch with this lady. So here you are.
Hi, Swati. I accepted your LinkedIn invite. I was
Yes.
nice.
See, it already happened, right? That is how it has to go. The proactive approach.
Proactive, yes. You have to be on the driving seat, right? You have to be navigating your career. Yeah. Good job,
So
Swati.
for all the listeners out there, as second speaker, I know Dr. Rupali, it has to be fixed between 12th and 13th, but tentatively I've put it 13th here, 13th March. So the date and time always we can negotiate depending on your convenience. And this is the second talk of the series, The Possibilities for a Science Enthusiast. And again, here's a great basic biologist, taking it to higher levels and actually getting to the people, the real people, and making it happen. Whatever, experiments we do in the lab with the hope of, making it useful for the mankind. That is the whole thing. And I think Rupali has really justified that in a big way. And so does Swati. Swati and Rupali, I think it will make a big combination. You people should... So, Dr. Swati, it would be great if you join, in our next session with the...
Sure. I would love to join, and I would love to hear from Rupali, and it's nice to be connected. It has been really pleasure speaking with you all, and especially Ravindresh. I think we kind of were on the same page for
here also. I
and
really enjoyed talking to you, and I hope that we can, meet you physically now. I mean, it's the online thing is fine, but we would love to have you here in our university someday.
Oh, yeah, we would try our best to pull them up at CUBB. Yeah.
Yes.
I am a foodie, and I would love to see Punjab. So yes, all goes there. Okay. Thank you, Manju. Thank you so much. This was a really great talk and a well time spent on Saturday, I think. For everybody, take home messages: do work hard every day as if you are working for yourself. I do see a message in the chat box, and which was like, I just want to point a little bit, I said something. Do you mind, Manju? Just one thing I want to
Oh, please go ahead. I'm not
I
my.
was just looking at the questions here, and the Ram Kumar asked a very good question. My question is that during your very hectic research schedule, how you manage stress, and especially when you are working for a long time and not seeing instant effect of your work on people's life.
Yeah.
So, Ram Kumar, managing stress is a everyday process. And what you are seeing at your age, it gets double and triple and quadrupled with age. A lot of things happen when we grow old. Family, kids, and so when you are yourself, what you are talking about stress, as I used to feel it that way too when I was in PhD. And I'm like, oh my God, there is so much stress of completing this and showing I have a meeting with the madam this next morning and I don't have the data. Do I have to keep on working till 5 o'clock in the morning to finish that? So managing stress, the way I used to do it, was in JNU, there was a yoga club. And I used to go every day in the morning, 7 o'clock, for just 1 hour yoga. And it used to just de-stress my mind for a little bit before facing my madam 9 o'clock meeting. I'm like, I'm going to take deep breaths today. And then the second thing is managing stress is, which I have learned over the period, is not to, like sometimes, when we respond, we react to the situations and we overly react. The situation is not that particularly complex. You can just take it as it is, but we make it bigger than it is because that's how human is, right? We think, oh my God, this is such a big problem. So take it as it is, and take some time to absorb it and come it with a fresh perspective. And I tell my boys, like, when you are stuck somewhere, just take a break and think it differently, and you will have some solutions. So there is always a stress. There is it's a everyday process and for everybody. But it's like how we react to it that makes a difference at the end of the day.
Yeah, that is very well said, Swati. So with this, I think we can conclude today's session. And yeah, I'm really happy to have Rupali and Swati together somehow. Dr. Ravindresh, thanks a lot. I'm really, impressed by your enthusiasm per se. So I'll be involving, more of you in all these, activities. So that's really something great. And I would really request Dr. Swati and Dr. Rupali to pass on the message. We can have more speakers like you guys, so that we continue the series. Like I'm trying my level best, to get the right sort of people together.
Sure, definitely.
Push these youngsters, way ahead and to generate some enthusiasm towards like everyday stress and everyday joy, isn't it?
Yes,
so
that
is true. Has to be spent. Yeah.
Good things happen when you are in stress. Let me tell you this way.
Okay, Swati then. Yeah. Bye. Thank you so much.
Bye. Thank you so much. It was a pleasure. Bye-bye.
Bye.
Sure. Bye. We'll catch up later. Okay, Dr. Rupali. So we'll be together sometime in mid-March, right?
Yeah. Looking forward to it.
Yeah,
Good
sure,
job, guys. Yeah.
Thank you so much.
Thank you. Have a great weekend. Bye.
You too. Bye. Bye.